Question 25. You are a researcher who wishes to isolate and remove a gene from a human chromosome and to incorporate it into a plasmid vector. It is important to use the same restriction enzyme to remove the gene from the chromosome and to open up the plasmid ring. Explain your answer. Restriction enzymes cut at specific sequences, so the same restriction enzymes must be used because it will produce fragments with the same complementary sticky ends, making it possible for bonds to form between them. Restriction enzymes is a protein isolated from bacteria that cleaves DNA sequences at sequence-specific sites, producing DNA fragments with a known sequence at each end. The main application of restriction enzymes are construction of restriction maps, construction of DNA fingerprints, recombinant of DNA technology, so rDNA technology. It is important to use the same restriction enzyme for removing a gene from human chromosome and for opening up the plasmid ring, because using the same restriction enzyme will generate same sticky ends in the gene as well as the plasmid. These sticky ends will form complementary base pairs, and thus the excised gene from human chromosome will get easily incorporated in the bacterial plasmid. Question 19. A drug becomes attached to the repressor protein and prevents the protein from binding to the operator region. Describe how this would affect the transcription of the LAC operon. LAC changes the structure of the repressor such that it is unable to bind to the operator site, which in turn allows RNA polymerase to transcribe these structural genes. The LAC operon is repressed by LAC-L, encoded by LAC-L. The LAC-L gene is upstream of LAC-ZYA and faces in the opposite direction. The repressor, LAC-L, binds to the operates sequence upstream of LAC-ZYA and prevents transcription of those genes unless the inducer molecule is present.